Great. Well, welcome to another episode of Systems Neuroscience and Complexity. I'm really excited to have Caio Seguin with us. Caio and I met a number of years ago at some Australian conferences when he was working with Andrew Zaleski, and he was doing really cool work thinking about communication models in networks and all these really cool, funky algorithms you could use to kind of think about this really interesting idea of information processing in a biological system. And so I think, that was the start of what has been really fun to watch, Caio. You're doing really great work now on your postdoc with Olaf, and I know you're about to come back to Australia. So this is, I think, a really great time for us to hear what you've been up to, what you're interested in, and have your ideas in the back of your mind, everyone, for cool things we might work on when he comes back in spaces that we can have discussions about. So really
Cool.
excited to have you. Feel free to take it away, share your screen, and off you go.
All right, thanks, Mac. Thanks for the introduction. Let me share the screen. Oops. All right. Can you guys see the PowerPoint?
Yeah.
All right, cool. So yes, I'm going to talk about my work on brain network communication. And this is going to be shaped according to a review that came out recently. And I'll put the link up in the end. Okay, please interrupt if you have any questions or any comments. All right, so I'm going to argue that network communication offers a way to capture, sort of provide a bridge between brain structure and function. And I'm going to start with two examples of that. So I really like this example, and apologies if you've already seen me provide it before. But here we have, on the left, a structural connectivity matrix. So this is the human connectome, macro scale connectivity mapped with diffusion MRI and white matter tectography. So these are anatomical connections. And here on the right, we have functional connectivity from bold fMRI. So these are patterns of statistical associations between time series, regional time series. And the point that this figure makes is that most functional connections are actually not underlined by a structural connection below them. So this matrix tends to be typically sparse, whereas this matrix tends to be typically dense. So this is pointing towards this idea that if you have two regions that are synchronized in time, and yet they do not share a direct anatomical connection, this synchronization between them must be facilitated by at least one intermediate region doing something between them. And if we look at a completely different scale, this is the paper by Michael Winding that came out in Science a while ago. And they've mapped the complete connectome for the fly larva. And this paper was beautiful. I loved this paper. So here is what it looks like. It's quite a weird thing if you are used to sort of MRI in the brain, the human brain. But this is what the connectivity matrix looks like. And what they did is they used a cascade model to simulate communication on top of this matrix. And they simulated the communication starting from neurons that have to do with sensory input, all the way cascading down to neurons related to motor control, so connected to muscles. And what they show here is that you have these really long polysynaptic paths that facilitate this sensory motor flow of information through this structural connectivity. Oops, sorry. And what both of these examples try to get at is that brain function depends on this polysynaptic communication that is constrained by the underlying structure. So my research asks this question: can we use tools from network science to model how structural connectivity might shape and constrain neurocommunication processes? This question can be approached through a lot of different lenses. So you could think about it from a perspective of information theory, like Joe does. You could think about it from a perspective of dynamic systems, like what you guys do. So in this talk, I'm going to approach it from the perspective of network science. So using tools from graph theory and network science to try to understand this. And later down the talk, I'm going to talk about how we might integrate and sort of bridge these gaps between different modeling domains, which I think is pretty exciting. Okay, so to start with some basic concepts, I'm going to focus on the human connectome. So we have here a toy human connectome. A walk on a graph is any sequence of nodes and connections that link to nodes of interest. So here we have I and J, and here we have a walk. And a walk doesn't impose any restrictions on whether nodes can be revisited or connections can be revisited. You can just do whatever you want on a walk. A path is a walk in which you can't revisit a node or a connection, right? So here from I to J, this is an example of a path from I to J. And the shortest path is the path that goes from I to J with the minimum cost. So if you think in terms of number of hops through the network, this is the shortest path from I to J. So you have 3 hops. So this is just some basic connections. And you can think of the sort of this kind of the length of the channel of communication going from short, the shortest path, to long, where walks tend to be longer. Okay, so what are the ideas that people have come up with to model these network communication processes in the brain? So there's kind of 2 main families of communication models. The first family is what I call routing protocols. So the idea is if you want to go from a source to a destination, you route the signal via a selectively accessed path. So here, a very direct path between source and destination. So this is kind of a routing scheme. But you can also think about it as a diffusive process. So if you start at a source, rather than sending it down a selective path, you just kind of diffuse this information around. So it propagates via multiple fronts until it reaches a certain destination. So you have routing protocols and diffusion processes.
Can I throw a quick question at you about the diffusion?
Yes, please, yeah.
Can you think about this one also kind of as a probabilistic routing? Like if you look at the strengths of the connections out as like a probability of a message going down that channel, then over the ensemble of running a probabilistic process like that, you get the same thing as the diffusion. Would that be right?
Yeah, so typically the way people have applied these routing protocols, it has had traditionally a more deterministic flavor. Whereas the diffusion processes have a more stochastic flavor. But you could implement a stochastic routing process where the decision is not fixed, but it is based on some sort of probability distribution. You could do that, yeah. Is that related to what you're asking?
Yeah, I mean, I guess I'll just say it seems to me like if you did probabilistic routing, that should probably basically give you the same thing as diffusion if you average over all the possibilities that you
over all the possibilities. Yeah. So if you think of a single random walker propagating through the network, you could think that yes, it is going to trace a single communication path. But the idea is that will tend to be much longer than these models that are based on routing, which try to deterministically find an efficient path between 2 regions. Does that kind of make sense?
Yeah.
I think I will provide some more examples that might perhaps clarify the distinction.
Yeah. No, I was just curious whether that third option was something people talked about as well and had already the same as diffusion.
So these are kind of very broad families. And in this next slide, I actually tried to summarize into a taxonomy all of these different ways of thinking about communication in networks that have been applied to brain networks. So we start with these 3 families. So we have the diffusion processes, we have routing protocols, and we have these parametric models that are hybrid in between the 2, which I'm going to explain in a second. So a family is what I'm calling this broad conceptualization of neurocommunication. Then you have a model, which is a specific strategy of how the signals are propagated or how you identify a path between regions. So for example, the classic routing protocol is just the shortest path between two nodes in your network. So what are the properties of communication via shortest path? So by definition, this would be the most efficient way to transmit a message in the network. And if you think of the metabolic cost of actually sending some sort of action potential down an axon, it would also be very energetically frugal. because you would minimize crossing gap junctions, and you would minimize the cost of propelling an action potential down an axon if you did shortest paths. The problem with shortest paths is that for you to know the shortest path between two nodes of a network, you actually need to know the entire network. So this is what I call that it requires global knowledge of the network. If you wanted to compute the shortest paths, you need to have access to the entire network. The kind of polar opposite of shortest paths is just a random walk. So in a random walk, each node, you make a random decision of where you're going to go next. That could be based on your connectivity weight, but it remains random. And it has the opposite properties. It would be very inefficient to send information that way. It would be energetically expensive, but it could be feasibly implemented in a biological system. All you need to know is who you're connected to. So this kind of provides this spectrum from decentralized strategies to centralized strategies. And there's a lot of different models in between. A lot of different things have been proposed. This is just a very small subset of what is actually out there. And one thing that we thought about is these three dimensions of communication cost. You have the delay, so how efficient your transmission is. You have the energy, so what are the metabolic demands of sending information according to a certain conceptualization of network communication. And you have how much knowledge about the network you need to know in order to route a signal according to a certain algorithm. So these like three dimensions of communication cost help us think conceptually about how these different algorithms might be plausible or not. So it offers a way for us to qualitatively assess these different strategies. And finally, what I call a measure. So we have family, is broad conceptualizations. Model is a strategy to propagate the signals. And the measure is actually how you quantify that signal propagation. And each of these models have many, many different ways in which you can actually quantify what's happening when you're sending this signal between regions. And these measures come in different flavors as well. They can conceptualize efficiency of transmission. They can conceptualize cost of transmission, how much information in a very sort of information theoretical sense in terms of number of bits you would actually need to implement a certain strategy. You can quantify resilience in terms of how many alternative routes you have between two different nodes. You can benchmark you can benchmark the efficiency of one measure versus the other. There's a lot of different things. So I'm not really going to get into the detail of this, but there's a lot of different ways in which these models can be used to quantify network communication. So I am going to explain in a little bit more depth two of these models, and I think that will help kind of fix some of these concepts. The first one is this work that I did in my PhD with Andrew Zaleski, is this idea of network navigation. So what if we could achieve efficient communication without needing to know the entire network topology? So the idea of navigation is that you send signals based on a measure of distances between nodes that you only need to know locally. So this is actually inspired by work in the 60s by Stanley Milgram, who was a psychologist, more famous for his work on authority and electric shocks. But he also did this very, very cool experiment where, and you might have heard of this, where he selected two samples of people in two different states. I think it's like Nebraska and Boston, so very far apart. And for the people in Boston, each participant got a letter. And they were told, you need to send this letter to a recipient all the way in Nebraska. You have their name, you know their address, and you know their job. And you need to route this letter to them. But the catch is that you can only forward the letter to someone you know on a first name basis, right? So this requirement for the first name basis maps a social network of acquaintances, right, throughout the country. So it's someone you know. And the idea is that you have to forward this letter, and the next person will forward it as well, until maybe you will get to the intended target. And the way people did this is that because they knew where they lived, they thought, okay, my cousin lives in Nebraska, so I'm going to send a letter to them because that will get it a lot closer to where it needs to go. So based on geographical distance, or based on actually job distance. So if you think, I don't know, I have a friend that is a dentist and I know that the target is a dentist, so I'm going to send it to him because he's more likely to know someone closer. So it's a very greedy strategy based on some knowledge of node distance in the network. So we kind of thought, okay, does this work in the brain? It was kind of like a fun question. And the brain is a spatially embedded network. So we thought, let's try to do it with Euclidean distance. So the rule is really simple. If the message is with you, you just propagate it to the neighbor that is closest in distance to the target. So here's a little example. Here we want to go from A to H. The shortest path from A to H is in red, like this, A, B, B, E, E, H. So that's 3 hops in the network. But the navigation path, you actually, instead of going to B, you go to D, because D is closer to H than B. So you go A, D, F, G, H. So that's the difference between the shortest path and the navigation path. So we thought, okay, how efficient is this compared to the shortest path? So we came up with this measure of the efficiency ratio, which is really simple. If your navigation path has 4 hops and your shortest path has 3 hops, the efficiency of navigation here is 3/4, so 75%. And then you can compute that for every pair of regions and you can average that. And this is what we are showing here in the y-axis. In the x-axis, we're thresholding our human connectome at different connectivity densities. And we can see for typical connection densities around like 25%, we actually get around 80% of the efficiency of shortest paths, only based on these very simple heuristics. Bearing in mind that shortest paths achieves its efficiency knowing the entire network topology of how 200 nodes are connected and all of the weights of all of the connections. And navigation only knows this distance between nodes. So we thought this was cool because it provided this kind of 3-way relationship between topology, geometry, so the spatial embedding, and communication. And with weaker assumptions about what information is available to nodes, we could send these messages with almost the same efficiency. So if we think about our little tag, we could say that navigation has a good transmission delay. It still has this problem that it requires you to know how far away your nodes are in relation to a target. And that is very hard to conceptualize how the brain would actually do that. But the point here is to show that you can come up with decentralized communication strategies that also work really well. You don't need to know the entire network topology to come up with a way to send information in an efficient way. There's proposals to how we can adapt the internet to send information like this instead of shortest paths. This is kind of where this line of research has more traction. So this is the idea of navigation. Another model that I really like is this idea of broadcasting. So this is more related to the diffusion conceptualization instead of routing. And here I'm going to think of broadcasting in terms of a particular implementation that has to do with communicability, which I'm going to explain what that is. So communicability proposes that if you're transmitting a signal, it's going to be transmitted via all possible walks between a source node and a target node. So what does that mean, all possible walks? So for walks of length 1, the signal that starts here is going to go to the direct neighbors of the source. So this guy and this guy, right? For walks of length 2, you're actually delineating all sort of 2-hop paths from the source. So 2 hops walk. So you can go from here to here to here. This is what a walk of length 2, here to here. But you can also go back to the source. So it's really all possible walks. And for walks of length n, the number of walks between source and target is given by sort of if A is the adjacency matrix, Aij to the power of n is then how many walks you have of step n. So if all walks contribute to communication, we can derive this formula that gets the communicability between two nodes. So essentially what you're doing is you're saying, yes, all walks contribute to communication, but walks that are really long become downplayed. So the importance of a walk is inversely proportional to its length. So here, the down weight is n factorial. And this is a geometric series, and it converges to the exponential of the adjacency matrix. So this is a measure called communicability. It's really used in complex networks, and I'm going to get back to that. I know that this is very abstract, but I'm going to show in a few slides an application of communicability to real data, and it's going to get a bit clearer how it works. But the point that I want to make here is that this strategy doesn't really mandate you to know anything about the network. You're just sending information indiscriminately. Since the shortest paths between two nodes is part of this set of all walks, in terms of the transmission delay for something to travel between two nodes, this would actually be the same as the shortest path. But this strategy is very, very metabolically demanding because you're constantly sending signals back and forth. So you could think in terms of our little 3 tag of our qualitative assessment. You could think that communicability achieves good delay. It doesn't require a lot of information about the network, but it's very wasteful in terms of energy because it's this kind of indiscriminate broadcasting idea. Okay, so hopefully these 2 examples kind of make it clear what the idea is. The last thing the last example is a biased random walk, which essentially they have a free parameter, and you can control this parameter to decide how much information you're going to endow your system with. So here you can think, so in this example, this is your network. You're starting from node 1 and you want to get to node T, right? These orange arrows denote the transition probabilities of a biased random walk. Here, we are not introducing any bias to the system. So this is just going to behave like a normal random walk. So if you start at node 1, a potential random walk that you could describe is something like this, right? You go around here a couple of times, then you come out this way, this way, this way, and eventually you get to your target. But if you tune this parameter with a target T in mind, you actually downplay the transition probabilities of connections that do not lie on the shortest path to the target, and you upplay the transition probabilities of connections that lie on the shortest path to a target. So here, in this case, you are already describing a more direct path to T. And if you put this parameter up to its maximum, you're actually implementing shortest path routing. So it's just a nice way to parameterize how much information you give your system. So they encapsulate in a single model this entire spectrum from routing to diffusion. And of course, in this case, the qualitative evaluation of the model totally depends on the parameter that you set. And what is really cool is that if you actually plot, so here on the x-axis, you have the parameter. So higher means more information into the system. On red, you have how efficient it is. So the cost of sending a signal in terms of the delay. And here you have how much information you're inputting into the system. And these curves don't really move at the same speed. So what you're saying is that you can decrease the cost of transmitting information a lot without actually endowing the system with a lot of information. So maybe there's a sweet spot for this parameter in which you could realize efficient message passing without actually requiring global information of the system. But this is a very new model and no one has really done that, but I think it's kind of cool. So these are biased random walks. Okay, so these was all I'm gonna sort of get into the details of the particular mechanics of each model. And now I'm gonna move on to what we can actually do with these models. So the idea is that you start with a structural connectivity matrix. It doesn't have to be a human one. It could be a Drosophila one. And then you simulate network communication according to a certain model. So that's a Seguin conceptualization. And you use a certain measure to quantify this propagation. So you have a model and a measure, and that gives you a communication matrix. So you went from a sparse structural connectivity matrix to a full communication matrix where you have a putative estimate of how every pair of nodes can communicate. And then with this matrix, you could do lots of different things. If you average it across rows and columns, you get a single value for a whole brain that tells you how efficiently that network can send information according to a certain measure. You could average it to look at a particular system of interest, let's say the default mode network. You could average it across rows, so you have how easy it is to communicate to that region. Or you could average it across columns, so how easy regions can communicate outwards. And then you can use these values to try to sort of predict some sort of empirical or observational data that you have. So here, I'm going to talk about some of these applications of these communication matrices, and also, at the same time, steps towards actually empirically validating these different conceptualizations of network communication. So the first thing that people tried was to model functional connectivity. So this idea of structure-function coupling, where you compute your communication matrix on structural connectivity, you have some sort of neural activity recording that defines a functional connectivity matrix. And the hypothesis is that if based on the structure, two regions can communicate, let's say more efficiently, you would expect them to be more functionally coupled as well. So in this paper by Gogny and colleagues in 2014, this is what they did. They used this measure of search information, which I'm going to get to, also going to explain a little bit later. But the gist here is that they use search information. So this is this matrix here, this triangular matrix. And they compare that with functional connectivity maps from bold time series, so resting states. And they saw this really nice correlation. So search information actually measures the difficulty of communication. So you get a negative correlation. So regions that have a harder time communicating, according to this measure, have lower functional connectivity. And what I want to stress is that this measure is really easy to compute. It is an analytical transformation of the structural connectivity matrix. And yet it goes a long way into explaining variance in functional connectivity. So even though it's really simple, and it's kind of this stylized idea that the brain sends these little signals along the network, it seems to have a nice sort of explanatory power. So another idea that people have played around with is try to correlate these measures of communication with a certain phenotype. So this could be, I don't know, some measure of intelligence, it could be a measure of task performance, or it could be between group differences, patients and controls. I think this is cool. I'm personally less excited about this research direction. So I'm not going to go into that, and I'm going to focus on two other applications that I find a lot more interesting. So one is this idea that you can map how a disruption to your structural connectivity matrix is going to impact the function of the system. So here a disruption could be a brain lesion, could be neuropathology, could be anything. So now you get a matrix of disrupted communication, and you can see if this correlates with some measure of function. I'm going to talk about a really cool paper that did this, that I really like. So the idea is that you can use this thing called DREADS, which is a way to pharmacogenetically manipulate the activity in a targeted region. So in this case, they had monkeys, and they used this to pharmacogenetically deactivate the amygdala of these monkeys. So essentially, you inhibit activity in the amygdala, and they call it a functional disconnectivity of the amygdala. It's still there, it's still anatomically connected, it's just inhibited. So what they did is they mapped functional connectivity before administering this intervention and after administering this intervention. And they had a structural connectivity matrix for the macaque. They computed a communicability matrix. And what they did is they removed the amygdala from the structural connectivity matrix, and they recomputed the communicability matrix now it's a disrupted measure of communication, right? So you have the one before and after. And what they showed is that here you're plotting the disruption to communication, and here you're modeling the change in functional connectivity due to the intervention. And this is in the amygdala, right? So you can see that based on this communication dynamics simulated onto the structure, you can predict how the functional connectivity of the amygdala is going to change. But what is really cool is that not only you can predict the local effects of this intervention, but you can also predict the global effect. So now you have functional connectivity between cortical regions that did not receive the intervention, and yet their functional connectivity changed due to the fact that the amygdala went offline. And you can still predict this change in the functional connectivity of cortical regions based on the simulation of how communicability would change if you actually lesioned the amygdala in the structural connectivity matrix. So I think this is a really nice and compelling kind of demonstration of how these models can be useful. Another thing that I find really interesting is this idea of predicting brain stimulation. So here, you could think, for example, of TMS, right? TMS is a non-invasive technique to stimulate the brain. But often what we really want is we want to modulate the activity of a region that is not accessible via the cortex. So let's say the amygdala, for example, some cortical region. And if you have this communication matrix, what it's telling you is if you have a particular target, this column of the matrix is telling you which regions you might want to stimulate to have a greater chance that the stimulus that you're inputting somewhere in the cortex is going to be carried over to the amygdala. And vice versa, a row of the communication matrix is telling you if you stimulate here candidate site A in orange, what is the effect that is going to have in the rest of the cortex. So I tried to investigate something along these lines of brain stimulation. And we did this study where we actually used invasive brain stimulation using intracranial EEG. So for those who don't know, intracranial EEG is essentially you insert this electrode, this sort of lead, sorry. It's kind of like a needle, and it has many electrodes along its length, right? And you can insert many in a patient's brain. Each electrode along the length of this needle can both stimulate, electrically stimulate, and record electrical activity at millisecond resolution. These are patients with epilepsy, and this procedure is done as a preparation for resective surgery. So they are patients that have drug-resistant epilepsy, and the idea is you insert these electrodes and you stimulate different brain regions with the goal of finding what is the source of the epileptic seizures in the brain. But kind of piggybacking on this clinical procedure, all this data was collected. And the data was actually collated by something called the FTRACT project into these really nice formats of N by N matrices. So the idea here is, for example, in this case, you stimulate here and you have all the other electrodes reading out the information. So you have the cortical evoked potential in a downstream region. So here, region 1, and you can record a significant response if this cortical evoked potential goes over a certain threshold. So in region 1, we observe a significant response. In region 2, this activity does not go over a certain threshold, so we don't observe a significant response. And the beauty of the FTRACT project is that they collated all of this information across, in the data that I use, 550 subjects, 29,000 individual stimulations, and more than 2 million intracranial recordings into these matrices that, for me, as a person interested in networks and not really very EEG savvy, was quite nice. So it's an N by N response probability matrix that tells you across all of these subjects, if you stimulate region I, what is the probability that you will have a significant response in region J? And they also have the median response amplitude. And because they did this across so many patients, they could actually get a matrix that had very good cortical coverage So these are the stimulation sites and the recording sites. They do tend to cluster more prominently around regions involved with epilepsy, because again, the positioning of the electrodes is to try to find where the source of the epileptic activity is, but still you get this really nice cortical coverage. So we try to think, if we compute our communication measures on a structural connectome, can we actually predict the propagation of this direct pulse? So here, this is a schematic. You stimulate here, you measure a significance response here. The most interesting case for us thinking about polysynaptic communication is when there's no white matter tract between where you stimulate and where you record from. So this must be some sort of polysynaptic propagation through the network. So what we did in this paper is we got 5 different measures of communication. So these are our candidates to try to explain this propagation. And they range from kind of routing, so shortest paths, to diffusion, so communicability, and kind of a random walk here. So the idea is that if we simply correlate the communication measures with the amplitude, we actually get a really nice correlation. So this is for search information, and this is the amplitude of the response of the pulse. And these are for pairs of regions that are not anatomically connected. So we can empirically stimulate one region, we can measure the average amplitude of the signal in a downstream region. There's no white matter tract between these 2 regions, but we can simulate propagation via the intermediate topology of the network, and we can then predict the amplitude of this response. So this is for unconnected regions. But what I thought was really cool is that even for connected pairs of regions, the communication model, so here search information, did better than just looking at the structural connectivity weight between 2 regions. So in other words, if you have regions A and B, they share a direct white matter tract between them. You stimulate A, your best bet at predicting what is the response in B is not only looking at that direct tract, but what is happening on the surrounding topology of the network is also important. And we're going to try to explain to you how with this little schematic. And this is really the last empirical result that I have. So out of these 5 conceptualizations of communication, I'm just focusing on 3 because I think they're the most sort of educational and informative. One is the shortest path. So here we have 3 targets, A, B, and 3 places we're stimulating, A, B, and C, and we are measuring the response in a target C. So if this stimulus is being propagated exclusively via the shortest path in the network, we would predict that the response after stimulating A would be stronger than the response after stimulating B and the response after stimulating C, because A has a very short path, B has 2 hops away, and C is 3 hops away. Okay. Search information, on the other hand, this measure that wins, it actually says, yes, the shortest path is important, but also the local topology of connectivity along the shortest path is also important. The idea, technically, sort of in technical terms, search information is defined as if you send a random walker between 2 nodes, what is the probability that this random walker will travel by chance via the shortest path between them? So if the path is really long, this probability will drop. But it will also drop based on the connectivity of the nodes along the path. So here, A has many connections to the rest of the network. So if you stimulate A, this signal has a propensity to disperse and not really stay concentrated along the shortest path. The next node along the shortest path also has many connections. B, on the other hand, the nodes along the shortest path have fewer connections. Here just 2 and here just 3. So there's less propensity for signal dispersion here. Whereas in C, the nodes along the shortest path have very few connections, right? So in this case, if this conceptualization of signal propagation is correct, even though A, B, and C are connected to the targets via the same shortest path, you would predict that the response after stimulating C would be stronger than the response after stimulating B and the response after stimulating A. because the local topology along the path is different. And finally, communicability, as I tried to explain, I don't know if it was very clear, but maybe this will help. It considers the entire network topology. So here, from A to T and B to T, we have the same shortest paths. They are connected in the shortest way by 2 hops. But A has many, many different efficient ways to go to T, whereas B has very inefficient ways to go to T. So communicability is trying to measure as a whole, are there alternative efficient routes between A and B, between A and T, sorry. So the idea is that if this conceptualization of communicability is what's happening, or is a better model of what's happening, we would expect a stronger response from stimulating A than stimulating B, even though they are the same shortest path away from the target. And this is kind of what we found. We found that generally search information and communicability were the best measures, which sort of signals to this more diffusive conceptualization of network communication as being the most predictive one, at least of electrical stimulus propagation. So I thought this was pretty cool. All right.
Were there any impacts of stimulation frequency on those results? Could you kind of pull those apart at all? Because I know that neurosurgeons love to kind of zap at this frequency, but sometimes they'll change that protocol up to find seizure hotspots and things.
Yes. So the short answer is no, because the strength, this F-Tracks data set, it has lots of pros and cons. The pro is that by collating data across a lot of individuals, these individuals were mapped across 20 different hospitals in the world. You can get this really nice brain coverage and this kind of idea of what happens on the average. On the other hand, you lose all of the specific details of the stimulation that was done for each subject. Different hospitals will stimulate with different amperage, for example. It's a clinical procedure, right? So the clinician is not interested in creating the cleanest research data set. It's interested in eliciting a response. So it is biased on where it stimulates. It's if it sees that a region is doing something, it will stimulate there again and again. It will up the amperage. So this is the downside of this data set, that we don't have any of this information. And other SEEG studies do have that, but then the downside is that they have to be focused on very particular pairs of regions. Like we're stimulating the hippocampus to see what happens over here, right? So the approach of the F-Track project is the opposite. It's like we're gonna average it all out, but we're gonna get whole brain coverage.
Okay,
Does
cool.
that get to what you were asking?
That's good. I thought it was something like that. It's I mean, I don't think we need to go into it too heavily here, but the small amount of time that I spent with the intracranial EEG data I played with during my postdoc, we talked about this on SFN. It's a bit of a wild west at the moment in terms of protocols.
I know.
And as you said, there's often clinical decisions being made. I just want for the students that are curious of these kinds of data, that it's great, but it's very hard to do this.
I know. And I was and still am anxious about that, sort of embarking in this SEEG world. But what gave me sort of confidence is that the first people to criticize the data and to bring up these limitations were the people from the F-Tracks project.
Yeah.
Look,
it's just wide open. It's just there.
It's like, this is what it is. We really tried to do our best and process this thing.
Oh yeah, no judgment intended. Yeah.
No, I know. But they were the first ones to say that this data is a bit wild. Like it is an average of a mixed bag of different things. The hope is that the average will kind of wash out some of these problems and will give you something that is still meaningful. But yeah, no, I agree with you that, you know, every modality has its problems, right? Yeah,
totally agree. No free lunch.
Just looking at a different modality. Yeah. Cool. So just some closing remarks. So I hope I at least convince you a tiny little bit that these models, even though they are so simple, and kind of stylized conceptualizations of what might be happening. They're very flexible in that they can model different things. They can model resting state activity. They can model propagation of stimulation. They can model deactivation of regions. They're very tractable because they are analytical transformations of a structural connectivity matrix. They're super fast to compute. And I think that they offer a nice interpretation of how the underlying structural topology might be related to a certain process of interest. I think there's a lot of models, and there's not enough efforts to compare and validate these models. Everyone wants to propose a new model, and then we will end up with this huge zoo of network measures. I think network neuroscience has suffered from this problem for a long time. And I think that studies trying to systematically pitch these models against each other to try to refine the scope of useful measures are important in the future. And I think the way to do that is to focus on data sets that are interventional, where you have a particular perturbation to the system, where you have a little bit more of a causal flavor of what might be going on, or alternatively, longitudinal data sets. I didn't talk about this, but another application of these models is to track the propagation of neurodegeneration, for example. And that's a very successful application. So moving the focus from just describing brain networks or between group differences to actually thinking about prediction of the effects of an intervention or prognosis of how a disease might progress is really the way to go. I think we have used network measures to describe the brain kind of to death, and now maybe we have to move on to a more interventional or longitudinal kind of approach. And again, these models are super simple, and they are for sure not the whole story. They really lack neurobiological detail. They can't really tell us about mechanisms of flexible information transfer. They can't really tell you why a neuron would send something down a particular path, right? And I think in order to answer this question, we actually need to integrate different modeling domains. The networks that I presented here, but also dynamic models. So Bataya has some work about this, flexible routing as an emergent property of dynamical systems. They have some really cool work. Maria Pope, who is a student here with Olaf, is doing some work on this area as well. People in Australia are doing work in Andrew's lab in this area as well. So trying to see if we simulate dynamics, do any of these models kind of best capture what the story that emerges? So I think this is a very exciting direction. And that's kind of it. All of this is in this review that we published with Olaf and Andrew. I took many years to finish that. So it was a work with the University of Melbourne, still in my PhD, and here is my postdoc. And Olaf and Andrew have been very patient with my, with my turtle pace. But yeah, if you want to know more, read the review, send me an email. And that's it. That's what I had.
Great stuff, Caio.
Yeah.
I think my introduction is ringing in my head. I couldn't be more impressed with, watching what you've done over the years and developing your ideas and really putting lovely kind of barriers and scaffolding both around them and underneath them. I still remember a number of chats we had over at HBM's, over poster sessions with these ideas. The fact that they've sort of become formalized now is really great to see, and I think that's something I'd encourage the students to be thinking about too as you move forward. It's a service to your field to think carefully about what you can get from any of these different methods, and taking this bird's eye view, you can say to yourself, you know what, none of my methods is perfect. Some of them are good for some things, some of them are good for others, some of them are really bad at certain things, so I probably shouldn't use it for that. But kind of understanding your tool or your craft in a way, I think is a really important feature of being a modern scientist. Joe, you've raised your hand, and I'm really curious to hear what you'd like to ask Caio.
I have. Thanks, Kai. That was really interesting. For a long time, we've been thinking a lot about how network structure constrains, impacts, or however you want to say it, the dynamics. And I think, just picking up on one thing Max said at the end there, the different measures might be useful for different things, as you showed. If you're looking at functional connectivity, this one's more related. If you're looking at, if I insert a pulse here, what reacts, well, then this measure is more related. So I think that's interesting. Along similar lines, on your last or second last slide, when you had the different conclusions and take-home messages, I was feeling like something that you kind of touched on a bit, but danced around in the end without saying, is it feels to me like we're kind of missing some theoretical results connecting the different measures of communication with the different things that they're useful for. You've got empirical results that you showed there. You've got intuition, but it feels like we're missing that last step of, mathematically being able to say, okay, this one is definitely useful for that kind of, this measure of structure is definitely useful for that kind of dynamics. This one's useful for that, and here's why. For me, it feels like that's a big missing piece here. Is that correct, or is there work that maybe you didn't mention?
So just to clarify, when you mean, this one is useful for that, that one is useful for this, do you mean the different network-based measures, or do you mean sort of the different?
That's right. So, you had a couple of slides from the end. You had a slide where you looked at.
I can put it up again.
The different, bring it up, and I can point straight to it. It's a good idea.
Yeah, it'll be easier. Is it this one? This one?
The slide before that.
That one. Yeah.
Yeah. So here in the bottom left. So what are we looking at here? So we're looking at.
So this is the correlation with the pulse amplitude, and here we have the different measures that we tried.
Yeah, that's right. So this is looking at how well these different measures predict the pulse amplitude after you inject the pulse.
That's right.
A couple of slides ago, you looked at how well. This one here, maybe it's not.
This one is when you disrupt a region.
Right. So this was about disruption, and then I think it's a few slides. Slide 15. You've got how well it correlates to functional connectivity.
Right.
So you sort of got coming back to that no free lunch idea. They each are better or worse at predicting several different kinds of dynamic things that you might want to measure.
Yeah.
And we've got that empirically here. And then on slide, was it 22, 23? You've got the really 21. You've got the really nice diagram in the bottom right of saying, okay, well, these different measures work in these slightly different ways. And so that can help us to start to get some intuition as to why they, are better or worse, or are better or worse at predicting different kinds of dynamics. So that's some good intuition towards that. But it feels like the next step after that is, can we go beyond the intuition and show this mathematically or theoretically somehow that, this measure of communication should be better at predicting that dynamics than others, and here's why. Does that make sense?
Okay. Yeah, it does. It's interesting because lately I've been actually thinking in the opposite way, like because what I struggle to realize how I could do your suggestion, because let's say I want to find out which measure is better at predicting the propagation of a stimulus, right? A pulse in this case here. If I want to do that from a theoretical angle, now I need to model this pulse using some sort of, I need a model of the pulse, I guess. But then if I'm only based on the model, how can I be confident that it would then translate to an actual empirical pulse? Do you know what I mean?
Yeah, I mean, it's the correctness of it obviously comes down to how correct the assumptions are in building the model. There's no doubt about that. I guess, and I'm not saying it's an easy thing to do. There's no way that's easy to do. But if it could be done, that's going to be better again than intuition.
Yeah.
In explaining.
No, I definitely get your point, and this is great because I never really thought about pick a certain flavor of communication problem, let's say post propagation, with an exogenous post, and demonstrate it theoretically or mathematically formally why this particular conceptualization of communication should be better at it. I never thought about that. I've thought more about the empirical side, but you're right. That's also good.
Anyway, just another possibility for future work.
Yeah. No, that's so cool. Yeah, thank you.
But I guess it sounds like there hasn't really been. Not only have you not done that yet, but it sounds like no one else has got those results yet either. So it seems like it's a big open opportunity.
Yeah. I don't think anyone's done it as far as I know either. So, yeah, cool.
Any questions from the students or anyone else in the audience? And if I saw a hand maybe there. Oh, Claire.
I have a question. It's a poorly thought out one. So if anybody else has a better one, jump in right now. All right, there we go. Caio, thanks. It was a really awesome talk. I really liked it. I guess, yeah, maybe my question comes a little bit from dissatisfaction with structural connectomes, and that I guess I find that side of imaging a little bit boring, and it doesn't interest me as much. But when I think about it, I always think that there's a lot of sort of missing ingredients in what we have there. And I guess I wondered a little bit about, I think it's a result that you've got here on the screen, where you say that even in these, these regions where they're structurally connected, that you still see that, the kind of structural connectivity matrix doesn't sort of explain everything, and that your models, add this extra little sort of secret sauce. And I guess I just wondered, from your perspective, knowing more about these and I guess coming more from the neurobiology of things, what do you think it's because we're actually missing just some really key ingredients in the structural connectome? Because I suppose, every time I think about it and think just how different these sort of connections are in terms of, say, just their myelination and the density of axons and things. And lately I've been reading a lot about glial cells. So sort of that's, all I think about at the moment. So, like a hammer and everything's looking like a nail, but there's even these amazing properties of astrocytes and like where they sit along axons and, if they're in the nodes and sort of how sort of densely packed they are, that completely will, will really influence sort of long range connectivity and things like that. And I guess I just wondered, for you, what do you think the missing ingredients are? And do you think actually, if we actually had more of that kind of neurobiological information in the structural connectome, that it would be explaining more of these things without the little secret sauce of the models?
I think it's a possibility. I agree with you 100% that these connectomes that we have are far from being perfect. We know this, right? We know that it has a lot of biases. There's problems with false positives, there's problems with false negatives, not to even get into the thing that you're saying about all these other neurobiological details that could be added into it, right? Myelination. I don't know. It's something I struggle with, but I try to, my philosophy is like, I know the data isn't perfect, but it's the best that we have at the moment, right? I'm just trying to use the best methods to get the best connectome that I can to then go with that. I think it's very important to keep those limitations in mind so you don't kind of delude yourself. But I think it's also equally as important to not let the present limitations sort of paralyze you and not let you do anything. This is kind of how I try to think about it. One of the things that I want to do is there's this preprint that has a very, very similar data set, but in C. elegans, and it uses optogenetic stimulation of individual neurons. And I don't know, I think that would be cool because if I could try to replicate the same results, now this is a brain that we know a lot of neurobiological information about. We know the cell types, we know when the neuron is born, we know everything, right? So this is one of those projects that's in the queue, but there's like 10 other things ahead of it, so I haven't gone to it yet. But yeah, this would be a nice way to test this idea, right? Is it really missing information, or are we onto something here? But I don't know yet.
Yeah, that sounds awesome. Thank you.
Yeah.
Other questions from the students in particular? Anyone have anything they'd like to ask? Well, for the sake of time, because I know we've lost Joe already, and I think there's a few other people that have some meetings to start. Why don't we put a pin in it here? But suffice to say, that was fantastic. Very much in scope for the kinds of things we want to be thinking about, and we often have really interesting discussions in-house about, you know, the idea of networks versus kind of brain states, and thinking about how to kind of map them together, build in neuroanatomy, and build models of these things. And so I think we should definitely continue the discussion once you're back. Let us know when you arrive.
I would love that. Yeah, for sure. For sure. Yeah. No, I know. As I said, I think I've lately started to think about how this line of research can be integrated with a more dynamic view of the brain. You know, once that project is you know, in the up for doing, you know, in the queue, then you guys will be the first ones I want to hear from, because you're the ones doing it. So the dynamic side of things. So it would be great to discuss.
And there are kind of like that graph you showed with all the different methods. There's so many questions in that space that need time and careful thought to get sensible answers. So the more the merrier.
Yeah.
All right. Well, join me all, please, again, in thanking Caio for his lovely talk, and look forward to seeing you soon, man. See what you come up with next.
All right. Thanks for having me, guys. Have a nice day.
Bye.